Okay, our second speaker is Olivia Pierce from BMS. Take it away.
Thank you, everyone. Thanks, Dima. My pleasure to come and talk about my work here. I'm a principal scientist at Bristol Myers Squibb here in Cambridge. So we talked a lot about small molecules binding to these single proteins mostly. So in this work, I'm going to switch a little over to a little bigger compound, not as big as what Romy talked about, but a little bigger that involves two different proteins coming together through these heterobifunctional degraders, particularly for IRAK3 degradation. So IRAKs or IL-1 receptor associated kinases are critical components of this TLR and IL-1 receptor pro-inflammatory signaling pathways. And in particular, IRAK3 shown here is thought to negatively regulate this signaling pathway. It was shown to be upregulated in specific tumor cells, and therefore the therapeutic hypothesis here is that the IRAK3 protein degradation will enhance the anti-tumor immunity overall in the tumor microenvironment. So I mentioned IRAK3 degradation. So why do we want to degrade IRAK3 instead of inhibiting as in small molecule inhibitors for a lot of therapies? IRAK3 is actually a pseudokinase, so it means that it's catalytically inactive in its nature state. So therefore, inhibiting IRAK3 is not an efficient solution for any therapy. However, because it's a pseudokinase, its overall structure very much resembles the kinase structures as overlaid here between IRAK3 and protein kinase A, and it has this very similar ATP binding pocket where the residues are quite conserved between typical kinases and IRAK3. So how and why do we want to degrade IRAK3? So in 2018, there was a patent from GSK that showed a pan-kinase degrader, which degraded 15 different kinases, including IRAK3, shown here. As you can see, the compound is quite big. Oops. Compound is quite big here. So I'm going to talk a little bit about the mechanisms of these degraders, or we call it at BMS ligand-directed degrader, or you can call it heterobifunctional degrader, PROTACs, a lot of names there, but through these LDDs. So what happens is if you look at the structure, on the right-hand side is the target binding moiety, which will bind to our target IRAK3. It is linking this target binding moiety through linker to the cereblon binding moiety, CBM. So the CBM will bind to cereblon, in our case, a non-native E3 ligase for IRAK3. So through this LDD, this IRAK3 will be brought to E3 ligase that is non-native in proximity. And on the other side of this whole colon ring ligase system is E2 ligase, which will transfer ubiquitins onto the surface lysine residues of IRAK3. And then this signals IRAK3 to be degraded and then shuttled into proteasome for degradation. So in this whole picture, the ternary complex formation between this target protein and the ligase through this LDD is very essential. But internally and also in public structure database, there has been no available ternary complex structure between LDD linking IRAK3 and cereblon. So in this work, I decided to use computational modeling workflow to first model these ternary complexes and also utilize free energy calculations. So there are several computational workflows to model ternary complexes out there. I used Schrödinger's workflow, which starts with protein-protein docking. So here we start with one protein, our target protein bound with TBM, and our ligase cereblon with CPM. There's no linker here, and it can generate up to 1,000 poses. What's shown over here is cereblon in green and basically a bouquet of our target IRAK3 docked in with protein-protein docking. For all of these individual 1,000 poses, we're going to try to attach the linker. It may not be always successful. So for the successfully linked conformation, then we will refine the interface of these two different proteins, including the linker region. And then we cluster based on the interaction fingerprints of the PPI. at this stage, I chose to cluster up to 5 different poses. And then these 5 different conformations are now going into this well-tempered metadynamic stage, which is used to assess the stability of these different PPIs of the ternary complex. And outside of the Schrodinger's workflow, I also used absolute FEP to assess the binding delta G of the degraders binding to these ternary complexes. So everyone knows metadynamics pretty good here. Like somebody mentioned earlier, the metadynamics let you travel the flattened free energy surface so that you can sample more evenly. In this particular case, as for collective variable, we use the RMSDs of the key residues interacting with the ligand. And the idea is that these PPI residues that are interacting with the degrader, if they move farther away from the starting conformation, that means the starting conformation of that ternary complex is unstable. So we don't want to choose that. So at the end, we choose the lowest RMSD pose as our top pose, because that indicates this higher stability of the PPI. So in terms of the degrader data set, I have from good, moderate to inactive degraders, where the modifications are all over the place, CBM, TBM, and linkers. And in terms of assessing the ternary complex models, I will look at the best model conformations and also how these ternary complex conformations distinguish the active and inactive degraders. I talked about the lysine residues being ubiquitinated for marking as the potential for degradation. So we will also look at how much these lysine residues are exposed after forming ternary complexes. And I wanted to also look at the bioactive conformations and strain energy of these degraders bound to the ternary complexes. So the team had arrived at this LDD13 shown here, which is the initial hit IRAK3 degrader, which has moderate potency. The first line shown here is IRAK3 binary binding of this compound to IRAK3, which is 16 nanomolar, quite good. But the degradation, DC50 and the depth of degradation are quite moderate, about 200 nanomolar to 70% degradation. The TBM utilized here is actually based on an approved cancer therapy that is targeting a different kinase, crizotinib. So the kinase selectivity of this compound is actually not so ideal. And it was also hitting a different neosubstrate in the neosubstrate panel. You see the linker region, it's quite flexible, few rotatable bonds. So we decided to rigidify it using a little bit more rigid spirocyclic linker, and then changed a little bit on the CBM side. Not super drastical changes, but few of them, which all resulted in overall improvement of IRAK3 binary binding and a little bit of improvement in the degradation. So this data set includes about moderately active to active degraders. So I took this set of degraders and then went through this modeling workflow that I just talked about. And interestingly, the top conformations that were coming out of metadynamics using based on the lowest RMSD, they were all common between these different degraders. So individual IRAK3 conformations are colored differently, and the cereblon is shown here in a fixed orientation. As you can see, they overlay very well. And again, these are all with different degraders. If you recall our degrader structures, the LDD13 shown in gray has the flexible linker, and the rest of the linkers, degraders, all remain the same, which indicated or implied that maybe there is a single ternary complex conformation that is efficient for degradation, which was very interesting. Then remember that when I went to the metadynamic stage, I took that 3 to 5 cluster representative poses from the clustering. So in this case, I took those. Now this is within the same degrader system. I took those and ran absolute FEP on the degraders. So the thought was, oh, the question was, the degraders bind more tightly to the more correct or crystal-like conformation? It turned out the answer is no. If I haven't blocking the screen, I should go here. So it turned out the conformation 3 shown here, which has the lowest metadynamics RMSD, which is our most crystal-like pose, actually has the highest absolute binding delta G for the degrader binding to that particular conformation. So here that was interesting to note, and then I started to think why. So when you run absolute FEP, you take your compound, and then these compound will go directly to your protein. In my case, the protein is not a single protein. It's a 2 proteins complex together. And when these 2 proteins come together, there should be some sort of reorganization, whether it is small side chain movement at the PPI or larger loop movement. But it is assumed to be exactly same across these different conformations, which are clearly different. So by assuming the same protein reorganization energy for these different conformations led to this disconnect or incorrelation of the metadynamics results and an absolute binding of free energy results. So that was kind of interesting and also disappointing. I thought I couldn't get more out of it because it took a long time to run absolute binding free energy calculation on this giant system. So then I started digging some more. What was interesting was there's this ligand strain energy from absolute FEP simulation. What was it? What I noticed was that conformation that was crystal-like or consensus among multiple active degraders had the low, very low ligand strain energy actually decreased compared to supposed to be decreasing compared to the solution conformation. So that was interesting. So I started to see this against multiple different systems. This is not the single system story. So I started to look at some more. So I first looked at the bioactive conformations and the ternary complex conformations of these degraders. It happened to be that for active degraders, the bioactive conformations were extended, and which is very similar to what you see in the ternary complex, because on one end, IRAC3 will bind, on the other end, cereblon will bind. But for the inactive degraders, bioactive conformations were all predicted to be more folded, in which case this does not allow in this conformation to have a ternary complex on top of it, because there will be significant clash between the target and cereblon. So for this inactive degraders to go to the ternary complex, it must have paying a lot of strain energy. So from the simulation, what I could notice was that you can look at the trajectory and then see the conformations. For active degraders in solution, both the extended and folded conformations were sampled. And actually, there were more of these extended conformations for the active degraders. Therefore, the average strain energy for the studied degraders were about 1.4. And actually, after I made this slide, I ran some more. It was actually below 1 kcal per mole. For inactive degraders in the solution, only the folded conformations were exclusively sampled. There was none of these extended conformations. So for these degraders to go from this very folded conformation to this stretched out conformation, they have to pay much higher strain energy. So this might be one of the reasons that these degraders are not so active. And maybe the active degraders have much lower strain energy that can contribute much more efficient degradation. We talked about dead degraders, inactive ones, so I'm going to talk a little bit more about them. So, but before going that, earlier I mentioned that our original LDD13 and the first set of degraders have this crizotinib-based TBM, which is not selective. So the team had surveyed some more TBM and arrived at this more selective imidazole pyridine TBM, which involved some change in hinge binding motif. With this, they incorporated different CBMs and linkers, and they were all dead. But I took these set of degraders and still went them through the modeling workflow. And what was interesting here was there were several ternary complex conformations that overlaid very well across multiple inactive degraders that from the metadynamic simulations. So this on the left-hand side is stable, and then this is also stable. So maybe there are multiple ternary complex conformations that are possible for these degraders that makes them inefficient for degradation. So then I also wanted to look at why is one ternary complex conformation more efficient than the other. Remember that the surface lysine residues of the target has to be ubiquitinated to be shuttled into proteasome for degradation. So there are 19 solvent exposed lysine residues on the surface of the pseudokinase domain of IREC3. For the active degraders, in the predicted crystal-like conformation, all 19 solvent accessible lysine residues were exposed. Whereas for the inactive degraders, 8 out of 19 lysine residues were buried, making them less likely to be, making the target less likely to be marked for degradation. So the team further optimized the TBM some more. Now we reincorporated the spherazole from the original AP series LDE13 like crizotinib-based compounds, reincorporated that with that into the IP series TBM. What that did was while we are having the change of the hinge binding motif, the overall trajectory of these two compounds are almost identical. The previous series that I showed for the dead degraders, it had phenol here, and then that actually had a totally different exit trajectory for these degraders to come out of the target. So their ternary complex conformations were not identical, but between these two different degraders and the rest of the series, with the change in TBM, we were able to preserve the same ternary complex conformation as well as improve the degradation potency much higher. So we did some more tweaks everywhere around the compound and then arrived at this LDD38 shown down here, which has almost a picomolar degradation potency and very deep degradation. We showed the IREC3 protein was degraded almost close over 94% in ex vivo studies as well as in vivo studies. This became our in vivo 2 compound. However, in vivo studies, there was some degradation disconnect between the amount of depth of degradation and the tumor volume reduction. So the program was terminated at the end, but I can now talk about it. Before ending, I wanted to touch on the relative FEP's usage on these ternary complex models. So this is with the very first set of moderately active to active degraders. Remember, they all had this identical ternary complex models, but if you look at the alignment of these ligands or degraders, they are not perfect. And in order to run FEP, at least for FEP plus, the atoms, the common atoms between two different degraders, they have to align on top of each other. Otherwise, they will be considered as part of your perturbation region. Your perturbation region gets bigger, it's not going to converge, and then I'm already running on this ginormous system. So then once I try to align these perfectly on top of each other, the ternary complex actually does not overlay on top of each other. Then what happens is that your the protein reorganization energy assumption again does not hold anymore. The protein has now covers much different space, deviates a lot. So perhaps that might be one of the reasons that I did not see any correlation between the delta G experiment and delta G of FEP. You can see also MUE is almost a polymer. Another thing actually to note here is delta G experiment is actually derived from the degradation potency, not from actual binding. We did not have any SPR or any direct measurement of the degraders binding to the ternary complex, although we did observe the ternary complex formation for these degraders. So that's one of the other things to note. Another set that I tried, relative FEP, involves some changes in linkers and actually some little smaller, much smaller changes in TBMs and CBMs. And that might actually sound like, oh, much bigger perturbation because you're changing the linker that might change the whole direction and not. But actually, there's a way to fix the TBMs if they are TBMs and CBMs in each target. So IREC3 and cereblon down here. And just sample the linkers, and then that drastically improved the alignment of the ligands almost perfectly. And then that will actually keep these entire ternary complex in altogether very identical. So you can still assume in this particular case, after that alignment, the same protein reorganization energy. So this, I think that is the reason that I'm seeing much improved results. R square of 0.44 is acceptable, but MUE is within the quite similar to the to what we normally see for small molecule FEP, a little worse. So with that, we show that these IREC3 degraders have the potency, selectivity, and PK properties in vitro and in vivo. But again, there was some biology disconnect that we cannot move the program forward. And the retrospective ternary complex modeling workflow that I used was able to distinguish these active and inactive degraders. And these active degraders likely have more extended bioactive conformation and have lower strain energy that might contribute to the efficiency of the degradation. We looked at the lysine residue content of the ternary complexes and also relative free energy calculations on these ternary complexes can be challenging because of the protein reorganization energy assumption and ligand alignment, but it's something certainly possible and then we do routinely. And that I like to thank Fareed over here, particularly doing a lot of medicinal chemistry work and helped me make slides. And also Augustina from Schrödinger helping me throughout the work, applying workflow and the organizing committee of the free energy workshop. Thank you.
Thank you, Olivia, for sharing this very interesting work. I think we have time for one or two questions. Is there a question? There you go.
Sorry, can you repeat the last part? Your question? Yeah. Yeah, so these are actually done for the same degraders, but just different ternary complex conformation. Yeah, and also after realizing that these absolute binding delta G for these degraders are under that assumption that same protein reorganization energy would be for each of these ternary complexes, I did not really trust any more of these absolute binding delta G for themselves. Yeah, but also besides the strain energy, there are multiple components that are contributing to the binding delta G, like interaction itself and the PPIs, which are all different in this case. So I think there are multiple components, again, going into absolute.
We're actually out of time. I'm sure Olivia will be happy to take any questions.
Thank you.
Post the session. Let's thank